All right, today we're going to talk about a microbe that's, well, everywhere, in the dirt, in the water, and sometimes with truly deadly results inside of us. It's a master of survival, a notorious hospital superbug. We're talking about Pseudomonas aeruginosa.
But let's start with a really strange question. Imagine you're a doctor, way back in the 19th century. You're looking at a patient's wound, and the pus is blue-green. What in the world could cause that? Well, believe it or not, that bizarre clinical sign was the very first clue that led to the discovery of this microbe.
And here's the culprit. Pseudomonas aeruginosa is what we call an opportunistic pathogen. You know, out in the wild, it's just doing its thing, part of the ecosystem. But when it gets a chance in someone who's vulnerable, maybe in a hospital or with a weak immune system, it becomes this formidable enemy, armed to the teeth with resistance against our best antibiotics.
You know, it actually took us a surprisingly long time to figure this bug out. It all started back in 1882 when Carl Gessard connected the dots to that blue pus. But for decades after that, the genus Pseudomonas was kind of a messy junk drawer for any bacteria that looked similar. The real breakthrough came in the 70s, when scientists used genetic tools to finally give it a proper ID card. And now, with whole genome sequencing, we can see its entire sprawling family tree in incredible detail.
So what makes this bacterium so incredibly successful? Let's dive into its amazing survival skills, its biological superpowers, if you will. First up, its diet. How many different things do you think this tiny organism can eat? The number is just staggering. It can use over 100 different organic compounds as food.
This adaptability is its true secret weapon. It's a metabolic master. I mean, it can eat everything from simple sugars all the way to chemicals you'd find in aviation fuel. It's also an expert mover with a little tail to swim and tiny grappling hooks to crawl across surfaces. And get this, it's a flexible breather. So if oxygen runs out, like deep inside a patient's lungs, no problem. It just starts breathing nitrate instead. And maybe most importantly, it's a fortress builder, creating these protective slime cities called biofilms, and we'll definitely be coming back to those.
So it can survive anywhere. But what's the one key trick that lets it jump from a puddle into a person? It all comes down to temperature. A lot of its environmental cousins, they like it cool. But Pseudomonas aeruginosa, it loves our body temperature, 37 degrees Celsius. It can even handle a fever. That ability is its golden ticket into the human body.
And once it's inside a host, it doesn't just hang out. Oh no. It comes locked and loaded with a whole arsenal of weapons designed to do as much damage as possible. One of its deadliest weapons is this thing called the type 3 secretion system. The best way to think about it is as a microscopic syringe. It's a 3-step process, and it's brutally efficient. First, it makes contact and latches onto one of our cells. Then, it builds this incredible molecular needle. And finally, it injects a cocktail of deadly toxins right inside, totally bypassing the cell's outer defenses. And what's in that toxic cocktail? Just pure devastation. Some strains inject a toxin called XOU, which is an enzyme so powerful it literally dissolves the host cell's membrane from the inside out. Others use XOS, which does two things. It paralyzes the cell's internal skeleton and then basically tells it to self-destruct. This isn't random. It's precision warfare at a microscopic level. And it doesn't just fight from the inside. It also releases weapons that attack from the outside. It secretes something called exotoxin A, which just grinds all protein making in our cells to a complete halt, killing them. And then it unleashes enzymes like elastase, which literally dissolves the elastic tissue in our lungs and chews up key parts of our own immune system, disarming our defenses before they can even fight back.
With an arsenal like that, it's really no surprise that in a hospital, where people are at their most vulnerable, Pseudomonas aeruginosa becomes an absolute clinical nightmare. I mean, just look at the range of infections it can cause. It's infamous for causing these awful chronic lung infections in people with cystic fibrosis. It causes deadly pneumonia in patients on ventilators. It gets into burn wounds, causes nasty eye infections for people who wear contacts, and can lead to life-threatening sepsis if it gets into the bloodstream.
But here's where this bug gets even smarter. When it decides to settle in for a long-term chronic infection, it doesn't stay the same. It physically changes its form, adapting to survive inside our bodies for years.
You can actually see this happen in the lab. When you first isolate it from a fresh, acute infection, say from a burn wound, it grows in these big, smooth colonies. This is its attacker form. It's mobile, it's aggressive, and it's ready to invade new territory.
But in the lungs of a person with cystic fibrosis, it transforms. It becomes mucoid,
which means it starts overproducing this slimy, gooey substance called alginate. This stuff creates a protective shield, this glistening layer that makes it almost impossible for antibiotics or our immune system to get to the bacteria hiding inside. This is the classic signature of a chronic infection. And then, after years and years of fighting off antibiotics, an even tougher form can show up, the rugose small colony variant, or RSCV. These colonies are tiny, dry, and all wrinkled. They are the absolute masters of building biofilms, hunkering down and clinging to surfaces. They're basically in full-on defensive mode, built to wait out any storm we can throw at them.
And this incredible ability to adapt leads us right to its most infamous superpower, its mastery of antibiotic resistance. It's like it has 3 different layers of defense.
So let's break down these layers. First, there's intrinsic resistance. This bug is just born tough. Its outer shell is like natural body armor, way less permeable than something like E. coli. Plus, it has these built-in pumps that just spit out any drugs that happen to sneak inside. Then you've got acquired resistance. This is where it learns new tricks. It can literally pick up resistance genes from other bacteria around it. It's like getting a software update for its defenses. And finally, there's adaptive resistance. This is when it hides inside those biofilm fortresses, where it can survive antibiotic doses that are up to 1,000 times higher than normal.
And get this, its defenses are incredibly sophisticated. It has a built-in alarm system. The moment an antibiotic starts damaging its cell wall, sirens go off. That alarm triggers a massive counterattack, flooding the area with a defensive enzyme called AMP-C that finds the antibiotic and neutralizes it. It's a perfect, targeted response.
So how in the world do you fight an enemy this smart? It's a constant, high-stakes race against evolution, and it demands a whole new generation of weapons from our side.
To fight back, scientists are having to get really creative. We're developing things like Trojan horse antibiotics, like cefiderocol, that trick the bacteria into thinking it's food. We're re-exploring phage therapy, which uses natural viruses as living weapons to hunt and kill the bacteria. And we're making smarter combination drugs that both attack the bacteria and shut down its resistance enzymes at the exact same time.
But ultimately, as this quote really drives home, the future isn't just about finding new ways to kill it. It's about getting smarter. It's about figuring out how to smash that biofilm barrier, all while being incredibly careful and protective of the precious few antibiotics that we still have left.
Which really leaves us with this one final critical question. You have to remember, bacteria have been playing this evolutionary game for billions of years. They are the original masters of survival and adaptation. The real question is, can our medical innovation evolve fast enough to keep pace in this fight?